That's right, Brody. Okay.
No, I mean.
He's talking about with the paper clips.
Yeah.
Yeah.
And we'll get back to that. We'll finish that.
Okay. So this is the replication fork here. Okay. And then it says during replication, the DNA molecule separates into two strands, produces two new complementary strands following the rules of base pairing. So then what's going to happen is we're going to build another side here and another side here.
And it connects together, right?
And it connects together by our base pairing rules. And if that fork keeps moving, it just keeps opening, and we add new nucleotides,
Will it ever disband? Like,
only when it's all done.
What do you think would happen if it disbanded before it was done? What do you think that might look like if that happened?
but then,
If it tried to open up, keep opening up, right? And if something got messed up along the way, what do you think might happen to the organism?
it would probably die.
It could die. It depends how severe. It'd have a lot of different problems that would happen. Okay, so this was...
Hold on one second. What do we call that when your chromosomes are messed up or when your DNA's messed up? What's the word that we use when your DNA's goofed up?
We'll do it start with.
M. What is it? Mutation.
Good.
Okay.
And then this was kind of basing on, goes with the other diagram, there's the new strand, the original strand, it separates into two, so that way it could duplicate and base pair with the original strand that it had. And these here, what are paired together are the nitrogen bases, the nitrogenous bases. And in case you guys remember, what does A pair with? The adenine, nitrogen, T, thymine, good. And what does the C pair with?
G.
Good, guanine. Good job. And then see we have the replication fork here, because that's when it goes from the opening, that's when it opens and splits into two, where it'll replicate. And then this is the DNA polymerase that kind of proofreads and makes sure that the replication that is going on is what it should be. Make sure that the nitrogenous base pairs are pairing correctly, because otherwise we would have, remember what would happen if it got messed up along the way? Does anyone remember? The DNA polymerase, it proofreads the bases to make sure that it's going correctly, the replication. Because remember, what do we call it when replication goes wrong? It's a mutation. Good job. So how replication occurs? DNA replication is carried out by enzymes that unzip, we call it, a molecule of DNA. And that's where you have the replication fork that we discussed earlier. It splits the DNA into a replication fork. Hydrogen bonds between base pairs are broken and the two strands of DNA unwind. So that's how it breaks apart and separates into the two strands so that it can copy itself. The principal enzyme involved in DNA replication is DNA polymerase. DNA polymerase joins individual nucleotides to produce a DNA molecule and then proofreads each new DNA strand. So that's kind of what we were talking about earlier. It joins, after it's been separated at the replication fork, the polymerase joins new nucleotides of a new strand onto the original strand to copy itself. And then it proofreads what it created to make sure that we don't have what we remember as mutations. Everyone got that? So now just kind of go over make sure that we got the right ideas. If you remember from before, in prokaryotic cells, DNA is found in remember prokaryotic cells, not eukaryotic cells. This is the organisms that are simpler, that have DNA. Where is it located for prokaryotes? If you remember. You can refer to your notes if you need help. Yeah, hey, the cytoplasm, good job. Remember that is for prokaryotic cells. DNA is in the nucleus of eukaryotic cells. Good job. The first step in DNA replication is blank. Is it producing new strands, separating the strands, producing DNA polymerase, or correctly pairing bases? Do you remember? What's the first step of DNA replication? How does it start? Remember what we talked about with what happens when a road splits into two? What would that look like? What does it have to do before it can perform DNA replication? What does it have to do at the very beginning? Separate. Good job. Yep, good job. We have to separate the strands first before we can pair new nucleotides and have a new strand connect to it. A DNA molecule separates in the sequence GCG AATT occurs in one strand. What is the base sequence on the other strand? Okay. Why don't we try thinking again, even so? This, what would G pair with, if we remember?
G.
What does a G pair with? Sorry. C. Good job. So it has to pair with its complementary base, right? So what do we think it is? B, you said? Yeah, it's B. Good job. Remember, it has to pair with its complementary base. In addition to carrying out the replication of DNA, the enzyme DNA polymerase also functions to unzip the molecule, regulate the time copying occurs in the cell cycle, proofread the new copies to minimize the number of mistakes, or wrap the new strands onto histone proteins. Do we remember? Remember, DNA polymerase has to try to prevent what? What do we call it when something goes wrong?
Oh, C.
Yeah, C, good job. Yeah, it proofreads the new copies to minimize the number of mistakes. It tries to prevent as many mutations as it can. That way the organism could be as I don't want to say perfect, but you want to have as little mutations as possible for the well-being of the organism. The structure that may play a role in regulating how genes are read to make a protein is the A coil, B histone, C nucleosome, or D chromatin. Does anyone have any guesses? Huh?
B.
B? Not quite, I don't believe. I don't know if it's...
C.
Yeah, it's C. Good job, Brody. The structure, the nucleosome has a role in regulating how genes are read. Good job.
End of section.
Huh?
End of section.
Yep, end of section. Yep. Does anyone have any questions about what we've been talking about?
No.
Change it up on you a little bit, Mr. Kramer. I think what we should do